At GenPath, we offer fluorescence in situ hybridization or FISH testing to physicians for better disease management. FISH can provide valuable diagnostic, prognostic, or therapeutic information. Let's walk through the process of how FISH testing is performed together. After a physician orders a specific FISH test or panel, the specimens are transported to GenPath, where the laboratory testing will be completed. For FISH testing at GenPath, we accept a variety of specimens, including peripheral blood, bone marrow, and formalin-fixed paraffin-embedded tissue. The appropriate type of specimen is determined by the particular cancer. The specimen is washed with fixatives and transferred into a centrifuge tube. The tube containing the washed specimen is inserted into a high-speed centrifuge, and the contents are spun down. Upon spinning the specimen, the fixed cells with intact nuclei will collect at the bottom of the tube. The fixed cells are transferred to a glass slide. The specific fluorescent probe ordered is also added to the slide. Fluorescent probes are short sequences of single-stranded DNA, complementary to a portion of the gene of interest and tagged with fluorescent markers. At the end of the technical process, the end user will interpret the fluorescent signals emitted from these probes. After the specimen and the FISH probe have been transferred to the slide, a cover slip is added and secured with rubber cement. Now that the slide has been prepared, the specimen must be heated for 12 to 14 hours for two important processes to occur: denaturation and hybridization. First, denaturation occurs. When heated, the double-stranded DNA helix within the nucleus of each cell separates into two complementary single strands. Then the process of hybridization begins. If the specimen DNA and the FISH probe are complementary in sequence, the FISH probe will bind to the specimen DNA and form a double-stranded DNA helix that is now fluorescently active. After 12 to 14 hours have elapsed, the slide is removed from the heat and transferred to the water bath to rinse off excess probe with buffers. Once the excess FISH probe has been washed from the slide, the counterstain DAPI is added. Now the slide is ready for imaging. Each cell within the specimen is scanned and digitally imaged. The FISH technician uses a fluorescent microscope to view approximately 500 cells in the specimen. For global cases, the FISH technician completes an initial review of each cell, discarding overlapping cells or those with incomplete hybridization, and classifying each remaining cell as normal or abnormal. For technical component-only cases, the automated FISH scanner performs the analysis, classifying each cell as normal or abnormal. Once all the cells have been digitally imaged and classified, the images are sent to a pathologist for final interpretation. GenPath enables external pathologists to perform the professional component of FISH testing through our web-enabled digital pathology service, StormPath. StormPath users can log into their account and easily access all patient FISH results. In StormPath, pathologists can interpret each cell in the patient sample tile gallery and reclassify cells if needed. After interpreting the FISH images, pathologists can sign out the case and a final FISH report will be generated. Utilize StormPath to provide FISH interpretation today. For a StormPath demo, contact GenPath